In this today's series of talks, the first topic is a role of ALK inhibitors in the management of non-small cell lung cancer, with a particular importance to the most important drug in the management of ALK positive non-small cell lung cancer, that is alectinib. So we'll be briefly walking through these headings: overview of non-small cell lung cancer, how do we test for ALK in non-small cell lung cancer, and when do we test, and what is ALK positive non-small cell lung cancer, and what are the approved with a landmark trials which have led to the approval of those ALK inhibitor, and finally on the most important ALK inhibitor, that is alectinib. So we all know lung cancer is a heterogeneous disease. It is broadly divided into small and non-small lung cancer, and non-small cell lung cancer constitutes roughly around 80%. And among them, adenocarcinoma, squamous cell carcinoma, non-small carcinoma, large cell carcinoma, not otherwise specified, they are further classified. So if you look at across the globe, roughly around 22 lakh cases of lung cancers are diagnosed each year, and around 18 lakh patients die due to lung cancer each year. If you look at the epidemiology in our own country, that is India, roughly around 72,000 cases were diagnosed in 2020 alone, and roughly around 66,000 deaths are due to lung cancer. So moving on to how do we look, we all know that particularly in non-small cell lung cancer and particularly in adenocarcinoma, majority of the tumor histologies are biomarker driven, and if we are able to pick up these biomarkers, we are able to drug them and we are able to improve the outcomes. So one such biomarker is a ALK. So how do we test for these ALK? So there are a broad variety of testing methods. It is FISH or immunohistochemistry or RT-PCR or next generation sequencing. The most commonly used and cheap method is immunohistochemistry, but because of availability of large number of biomarkers in non-small cell lung cancer, if we just go for each marker with a single test, probably we will deplete the tissue. So recent guidelines do suggest, instead of going for a single marker test, go for a broad panel testing such as next generation sequencing, which will not only include ALK, it also includes various other markers. So what ALK positive disease? We all know that ALK positive roughly constitutes around 3 to 8% depending upon the geography. It is mainly commonly seen in non-squamous histology of never or former smokers. Usually these patients are younger patients and more common in females, and these patients usually do not harbor either EGFR or KRAS. So if you look at the data from India, it is varies from 2 to 13% depending upon the studies. On an average, it's roughly around 3%. So what are the available molecules to manage metastatic non-small cell carcinoma, which is addicted to ALK gene? So first trial that has shown a significant improvement in progression-free survival in these patients is a Profile 1014, which looked at role of crizotinib compared to then standard of care, that is chemotherapy. So this trial has clearly shown that progression-free survival has almost improved from 7 to 10 months with the use of crizotinib, with a hazard ratio of 0.45, which has led to approval of crizotinib in the first-line management of metastatic non-small cell lung cancer. If you look at the overall survival, it was not statistically significant. However, it has shown that it tends to favor upfront use of ALK inhibitor compared to chemotherapy upfront. If you look at the toxicity profile, it is quite manageable toxicity. Majority of the toxicities are of grade 1 or grade 2. Grade 3 toxicities are usually to the tune of single digit, except for transaminitis, which is seen roughly in around 14% patients in crizotinib compared to chemotherapy. And another important thing that may patient complain is a visual changes that may be seen in 70%. However, grade 3, grade 4 visual symptoms are very rare. If you look at the clinical activity of crizotinib in brain metastasis, because this drug is usually pumped out by P glycoprotein, the CNS penetration and the CNS activity of crizotinib is less. However, in comparison to chemotherapy, crizotinib showed a numerical improvement in intracranial disease control rate to the tune of 40% at 3 months compared to 31% with chemotherapy at 24 weeks. If you look at another important molecule which was later approved in the management of metastatic all-positive non-small lung cancer is ceritinib, which was tested in a phase 3 trial, that is ASCENT-4 trial, which again looked at role of ceritinib compared to standard of care then, that is chemotherapy. Again, it has shown a dramatic improvement, almost doubling of PFS from 8 months to 16.6 months. If you look at the patients without brain metastasis, it has improved from 8.3 to 26 months to the tune of 3 times. But those who with a brain metastasis, the improvement was marginal from 6.7 to 10.7 months. But biggest problem with the ceritinib was when it was used at a dosage of 750, adverse events were very common, and adverse events leading to dose adjustments were to the tune of 70% in the ceritinib arm. Later in ASCENT-8 trial, they reduced the dose from 750 to 450 mg after low fatty meal, and they have shown that with a similar efficacy, however, with a reduced toxicity, ceritinib can be used at 450 mg after low fatty meal. The most common side effects of ceritinib were GI toxicities, particularly nausea, vomiting, and diarrhea. Apart from that, fatigue, pain, or insomnia can also be seen in these patients who take ceritinib. Similar to crizotinib, again, it is actively pumped out by P glycoprotein. So again, the availability of ceritinib in central nervous system is low because of the same. So similar intracranial response rates were seen with crizotinib and ceritinib. And also the first site of progression in both crizotinib and ceritinib were roughly around 46% and 42%. So another important drug which has recently got approved again in first line setting in management of metastatic lung cancer is a crown trial, that is lorlatinib. You can see the curves are separated markedly. The magnitude of benefit is huge, and median progression-free survival is not reached. And even at 3 years, 63% of the patients have not progressed on lorlatinib. So that is a big win for lorlatinib compared to when it was compared with crizotinib. So, however, it comes with a lot of toxicities. It is not an easy drug to digest. Grade 3, grade 4 adverse events were to the tune of 75%. That means that 3 out of 4 patients do develop a grade 3, grade 4 adverse events, and roughly around half of the patients do require a dose interruptions to manage these side effects. And important side effects with lorlatinib are weight gain, alteration in lipid metabolism, transaminitis, and also central nervous system toxicity, particularly mood changes, cognitive effects, and speech effects. They are of particular concern. So moving on to another important drug, that is alectinib. It is a highly selective to ALK4. So the safety profile, it improves the safety profile. It is highly potent, and it acts on multiple resistant mutations, and also it has a very good CNS penetration, which has led to improved efficacy of alectinib. So there are various trials which looked at the role of alectinib. We have a phase 2 trial, AF001JP, JLX, conducted in Japan, Alex trial, and also Alecia, which was conducted in Asian patients. So the main study, the Alex phase 3 study, which again looked at alectinib 600 mg twice daily versus crizotinib 250 mg twice daily. So if you look at the baseline characteristics, these also included a patients who had a brain metastasis, roughly around similar number of patients had a brain metastasis, and also they were regularly looked for evolution of brain metastasis in both the arms. So if you look at the primary endpoint, that is PFS, again it has shown that there is a significant improvement in PFS from 10.9 months to 34.8 months with alectinib compared to crizotinib. Nearly 3 years freedom from progression if patient is on alectinib. If you look at the subgroups, it favors across all the subgroups, be it a less than 65, more than 65, Asian, non-Asian, except for active smokers. it was favoring across all subgroups. If you look at the secondary endpoint, PFS by CNS metastasis, those who did not have CNS metastasis, the PFS was to the tune of 38.6 months. And those who had CNS metastasis, PFS was 25.4 months with alectinib. And the risk of disease progression, we all know that ALK positive patients do fail at CNS. So what are the incidence of CNS failures when they put on alectinib? It was to the tune of 10% with alectinib compared to the tune of 40% with crizotinib. And the curve seems to almost plateau even beyond year at around 10%. So if you look at the response rates, they were almost similar. But the important thing is the depth of response. The depth of response with the alectinib is much more compared to the crizotinib. More than 75% reduction was seen in almost half of patients compared to 1 in 4 patients with crizotinib. So updated 5-year OS trial, it was immature data. 50% of the OS events had not happened, but it was showing the trend towards improved overall survival with alectinib compared to crizotinib. So the 5-year survival rates with alectinib, if you look at 1 year, they are almost similar. But as we move towards 3, 4, 5 years, there is a difference in between alectinib and crizotinib. But 5 years, 62% with alectinib were alive compared to 42% with crizotinib. So updated safety profile, it is almost comparable with crizotinib. The few things of concern is with alectinib probably is rising CPK levels or transaminitis. Usually they are with dose interruptions, they can be easily manageable. And apart from that, very important thing when we compare alectinib versus ceritinib, which is most commonly used, is the drug interactions. Alectinib is almost as a very minimal drug interactions compared to ceritinib. So even clinical guidelines do suggest alectinib or lorlatinib as the preferred category 1 recommendations. What makes alectinib as the ideal drug for first-line management of metastatic non-small cell lung cancer who are ALK addicted? It has demonstrated a very good PFS. It is effective in CNS metastasis. It has a manageable toxicity, and 5-year survival rate is more than 60%. It is well tolerated. That makes alectinib one of the ideal drug for upfront management of ALK positive non-small cell lung cancer. These updated ALEX data confirm that alectinib shows a superior PFS, longer duration of response, impressive CNS efficacy, and also very good tolerability in ALK positive non-small cell lung cancer. With this, I conclude my talk. Thank you.
So it's